Rothen Rutishauser from Fribourg.
I'm already done. I really appreciate that you are still here, or maybe you already expected the first speaker of the next session. Anyway, I will try to hurry up. I also need a coffee. I would like to talk about something very different, as we already have heard during this morning session. I would like to show you how we try to target immune cells, and we have a special focus on the human lung because we have a huge surface which is easy accessible via inhalation. We can, by engineering the nanoparticles accordingly, we can also target the various immune cells which are present in the respiratory tract. And it has already been shown by some first in vivo experiments that such nanoparticles can either induce or suppress the immune reaction. And by inducing an immune reaction, it is also possible then to work on a vaccination approach. So our interest was really to do a lot of fundamental research to gain a fundamental understanding about how these nanoparticles can be designed to target and interact with the immune system in the lung. So here you see some images. This is a scanning electron micrograph of a human lung with a macrophage which is present at the apical site. And the main function of these macrophages is to engulf inhaled particulate matter. And on the lower image you see, now I'm also struggling here, so you see an immunofluorescent staining of dendritic cells shown in blue and macrophages shown in red, which is very numerous in the lung parenchyma of mice. It looks very similar in the rat lung. So according to this, we have established a system, a co-culture system, where we have implemented these two immune cells, macrophages and dendritic cells, which are derived from human blood monocytes, and we combine it with epithelial cells. It's also like Lego, so we can use different primed immune cells, we can use different epithelial cells. We have also started now to incorporate diseased cells, because if we apply nanoparticles, we usually try to treat a diseased person and not a healthy person. So we have now established a gold nanoparticle library to study the effect of surface charge on cell targeting. The gold nanoparticle, the gold core, was covered by different polymers to make them colloidally stable. We have used polyvinyl alcohol, or PEG. We also used a combination of the two polymers, and within this polymer we have incorporated here a fluorophore, because it is then easy, of course, to use a lot of fluorescent techniques to visualize and quantify the particles. We have made sure that these particles are stable over 24 hours in complete cell culture medium at 37 degrees. And then we have assessed the toxicity of these particles, or the uptake of these particles, by a so-called air liquid cell exposure approach, where we try to mimic the inhalation of these aerosols in vitro. You see here these drops which are generated, and then these drops sediment homogeneously on the cell surface of the lung cells, which can be cultured at the air-liquid interface. So I will only show you two examples, two particles, because of time, negatively charged particles and positively charged particles for the PVA, polyvinyl alcohol labeled nanoparticles. So we have deposited these particles on the co-culture system, which I just have shown you before. And after we have incubated, post-incubated cells for several time points, we have now applied a new procedure by detaching the cells with a method which we have just published now, and afterwards these isolated cells have been analyzed by flow cytometry, so we could quantify the fluorescence inside the cells. So these are the first results. You see here polyvinyl alcohol labeled or coated nanoparticles, negatively charged and positively charged, and here on this graph, you see the different cell populations. So we have clearly shown that for positively charged particles, we see more uptake in macrophages in comparison to epithelial cells and dendritic cells. And here we also see more uptake in the case of positively charged particles in comparison to the negative charge. So already by varying the charge, we have the possibility to target the specific cell populations in our co-culture model. We were then, of course, also interested in targeting the dendritic cells because these are the key cells for the development of allergic asthma, and also if we aim to go for a vaccination approach, this is really the key cell. And we have done this by attaching covalently an antibody to the surface of the polymer. This is a DC sign antibody, which has a respective receptor on the dendritic cells. And you see here the quantification clearly showed that we could reverse the targeting, so the targeting was much more pronounced for the dendritic cells in comparison to macrophages and still epithelial cells, which had a minority of particles inside. We also did a repeated exposure, and this also is in agreement with the study shown by Janos, that we also should do some repeated exposures, because this is also, if we apply nanomedicine in the future, we might repeatedly expose the patients to these particles, and we have evidence that over a longer time, these particles, these positively charged polyvinyl particles, can induce some reaction. We have assessed apoptosis, and we see that after 3 days, the positively charged particles can induce early apoptosis. However, this is not because of the gold nanoparticles, this is because of the polymer. So we also did this control experiment to really know which part of the particles is responsible for this apoptotic induction. The next step, of course, was then to also try these different charged particles in vivo. This has been done in collaboration with the University Hospital in Bern, and they have assessed the T-cell stimulation in the draining lymph nodes of particle-instilled mice. Here you see a dendritic cell which engulfs nanoparticles in yellow interacting with a T cell, and the T cell reaction is then shown here on this graph, and you see that only for positively charged particles, but not for the negatively charged and the polymer alone, we could see a significant increase in T cell proliferation. So taken all these results together, I really would like to emphasize that we should maybe also focus still a little bit more on a thorough particle synthesis and characterization. We have heard about agglomeration in the pristine bulk suspension. We also have the possibility to use different cells from different species to compare pig and human cells before we go into in vivo assays. And by applying this approach, we have really shown that the particles which we have used are stable in cell culture medium, and by applying the different charges, we could already direct the uptake a little bit significantly in the cultures by showing that more positively charged particles were taken up by dendritic cells if we attach a specific antibody to the surface. However, we also need to consider that maybe repeated exposure can induce some adverse effects. We also could show in vivo that the positively charged particles induced an enhanced T cell proliferation compared to other charged particles. With that, I would like to close, thank the BioNanomaterials group in Fribourg, but also the team which did the work which I just presented. I would also like to thank the Swiss National Science Foundation for the funding and you for your attention. And now let's maybe discuss later, afterwards during the coffee break. Thank you.
Thank you very much, Barbara. And we are right in time to close this session. Thank you very much for all speakers, all colleagues who came. And this is the end of this session. Thank you.